How many butterflies do you know? The monarch butterfly, plus maybe a few others. There are as many as 20,000 species of butterfly around the world, which have been cataloged over the years by a countless number of scientists and amateur collectors. But now that DNA sequencing is available, biologists have been able to double-check their work, and the family tree of butterflies has been given a Jerry Springer-level shakeup. In the 1740s, the first group for studying insects formed in London as the Society of Aurelians. Aurelian was an old name for butterfly enthusiasts. Moses Harris, an engraver, had made progress in representing vibrant colors with printing inks. He joined the society as a secretary. In 1766, he published the Aurelian, his definitive book of butterflies. Over the years, hobbyists have pinned these beautiful bugs to boards and mapped out a massive list of butterfly species. Margaret Fontaine became a notable scientist for her work on butterflies. In 1912, she was invited to join the Linnaean Society, which had only recently opened up to women, and was one of a few members in the Royal Entomological Society. Unlike other collectors and dealers, she would travel the world and raise butterflies on her own. Reportedly, she was found dead on Mount St. Benedict with a butterfly net in her hand. Her collection of 22,000 butterflies was open to the public.
When the collection was left to the museum, it came with a sealed black tin trunk, not to be opened until April the 17th, 1978. We discovered that there were 12 handwritten volumes, a journal describing her life from the age of 15 until just before she died.
When her diaries were made public decades later, she became iconic. As a child, the famed author Vladimir Nabokov started collecting butterflies. In the 1930s, he would write one of his Russian short stories about a butterfly collector and dealer. This would later be translated to English and also titled The Aurelian. Nabokov was committed to the role, volunteering at the American Museum of Natural History, and later continuing to travel the US and serving as a curator for butterflies for Harvard Zoological Museum, though it isn't clear how formalized that was. He sometimes mused that it could have been his career path in a different life. After his death in 1960, Nabokov donated his butterflies to the Cornell University Insect Collection, where it's available to students and researchers today. There's also a good amount of information about it online. A butterfly-focused pop science book, Nabokov's Blues, inspired researcher Naomi Pierce to give Nabokov's South American butterfly research a new look with DNA technology, and she published a paper agreeing with his theory that these species arrived in the Americas in 5 waves over the Bering Land Bridge. The paper cites 4 Nabokov publications between 1944 and 1945.
We've been able to validate many of the things that Nabokov thought. He's been right incredibly often, although he's also been wrong some. For example, in terms of rightness, his classification of the cardinal blue turned out to be spot on. In fact, they're the most distinct thing in the whole group, the subspecies he named, and should probably be a species if anything in the group is a separate species.
But the thing about DNA research is not every reveal is going to be so validating. Even before genetic testing, you can see wiki pages about butterflies are riddled with synonyms and former species, which got merged into others. What makes it difficult to classify these butterflies?
These two butterflies, even though they look very similar, are actually from different species. And in fact, if you move down the line here, this is actually one species with different color morphs, and this is a separate species with the same color morphs.
Male and female butterflies of a species often look totally different. And then a species may include multiple morphs or forms. So what are morphs? If you're familiar with ants, you know that a colony is one big family, but they're distinct worker, soldier, drones, and other ants, which, at an eye's glance, look completely different from each other. Depending on the genetics, epigenetics, and biochemistry of the colony, the queen will give birth to more workers or more soldiers. In butterflies, morphs often have different coloring. In this one family of South Asian butterflies, there is 1 male morph and 3 female morphs which look nothing alike. In this species, the colors are regional, mimicking toxic or undesirable butterflies in that environment. As recently as 1995, scientists warned against mixing the eastern and western populations of monarch butterflies. The eastern butterflies make a long migration, and the western ones go between the Rockies and the Pacific coast. By the mid-2000s, the genus got genetic testing. Now we know there's not much genetic difference between the two populations. It's believed to be a difference in gene expression. In 2004, an analysis of butterflies in Costa Rica found that the 2-barred flasher was at least 10 cryptic species hidden in plain sight. Later analysis of mitochondrial DNA suggests the number should be closer to 3 to 7 populations. In the Amazon rainforest, the clearwing or glasswing butterflies were sometimes thought to have between 7 and 28 different species. These have been rearranged based on genetic discoveries. Butterflies had a lot in common and produced hybrids after they had split off. But ultimately, some species had chromosomal changes, which made them incompatible with the other species, and they became isolated. Researchers at the University of Texas have found 2 so-called cryptic species in skipper butterflies using DNA sequencing. So what we want is a global sampling of butterflies, and then we'll have it all figured out, right? Well, people are working on that, but it's more complicated. In 2019, scientists finished collecting sequences for all of the butterflies in Canada and the US, about 800. But that's not as many as are known to exist in Mexico, and a fraction of those known in South America and Asia. In Europe, 1,000 genomes were completed this year, and a project covers almost all of the UK and will extend to 11,000 butterflies and moths across Europe. Even after making a genetic reference, there's the actual work of conservation, like this project in San Francisco, which is restoring the habitat of an extinct butterfly with a close relative.
Biologists from the Academy were able to identify the closest living relative to the extinct blue cerces. Combining sophisticated genetics with matching samples from their own collection, the results pointed to a species known as the silvery blue.
In fact, we sequenced the complete genome of 3 different subtypes of the silvery blue butterflies, so we could say which one was closer.
There are also parts of the genome which we don't understand yet. The mimicry of these butterfly species was traced to part of one chromosome. The initial interpretation was that the genes were physically close together, so they would be inherited together as a so-called supergene.
Supergene, a long stretch of DNA that contains many smaller genes. In the supergene, individual genes are immune to the random mixing and matching that normally occurs.
In 2024, 2 research groups instead pointed to a long, non-coding RNA which overlaps with this gene and produces microRNAs. There's not enough time to get into non-coding RNA, but what was once called junk DNA is on its revenge arc these days. Hey everyone, I'm a real human person trying to glue multiple articles together into one coherent story. If you learned something new, like and subscribe to help this channel grow. Thanks for watching.